Hey, tell me what you have here.
So this summer, I looked at the thermal effects of terahertz band radiation on the heart tissue. So we were looking at the terahertz band because it allows for faster communication generally, right? It's, you know about like 5G, you know how fast it is, right? You know how people were like afraid of it, like, oh, it's gonna harm you, it's gonna, but it really wasn't, right? Because people tested it, people figured out what's the safe limits, what's the safe power, right? What's the safe frequency? So that's kind of what I'm doing, what I did this summer. I looked at the terahertz band, and I determined how is it safe for the human body, right? So what we did, we used we used COMSOL Multiphysics, which is a physics software, to help us model how the waves actually propagates within the heart and how it heats it up. We're only worried about how it heats up the heart because terahertz band radiation isn't ionizing, meaning it won't break apart the molecules in your body, and it's only going to heat it up, right? So our goal was to ensure that terahertz waves are safe in the human body before implementation, and our focus was the heart. So for our lab, we basically modeled if we had a transmitter within the heart, right? And we built a heart model here. We have a 2D model and a 3D model. We just modeled how the waves actually propagates within the heart. Like we had we ran at different times with different frequencies, different powers, right? All those different variables.
You found the gigahertz work better?
I mean, I like this example, like what my PhD mentor, my lab said, right? Like if I spoke really low, right, but I spoke really fastly, right? Like if you're right next to me, you could hear me, right? And you would get a lot of information quickly, right? But if I spoke loudly and spoke quickly, right, you would get a lot of information, but you'd be able to be further away from me and hear me.
Oh,
right?
I see.
That's like how it is with power and frequency.
So you're giving up a low frequency. Is it a low frequency or?
I mean, we looked at it all. We didn't just look at one thing. We're just making sure we looked at everything. Because what our lab did, right, was to give other people data to determine how the devices could be safe in the heart, right? Because what's considered like unsafe is a temperature change of 1 Kelvin, right? Right, because like.
Okay, I see. So if it gets too hot, then it's an unsafe temperature. Is that correct?
Because we can't say if it's safe or unsafe just based on our own data, right? Because if you left the transmitter on for like a day or two, right? Eventually it would get hot enough, right, to actually damage the heart.
Correct. Yep.
But like let's say like a pacemaker, right? It's not always on. It's pulsing, right?
Pulsing. Okay.
Right. It's in like the heart.
Yeah.
I like that though. Very smart gentleman.
First group of presenters, we appreciate learning all about what you did this summer. Right now, we're going to take about a 7 to 8 minute break so we can switch over and get ready for the second group of presenters.
Neza.
All right.
Neza?
So
Neza.
Neza.
Yeah.
And then
All
we'll
right,
come
my
back
name is
to it.
Kenneth.
Thank you.
Kenneth.
All right.
You did a good job. You're a little nervous, but you did good.
Yeah, I mean.
It's okay.
No, it's fine. All right, but thank you. Thank you for listening.
You have good knowledge, though. I like the knowledge. That's the most important thing.
No, but thank you for listening.
No, I have to listen because how would I learn if I don't listen, right?
No, exactly.
So I have to listen. I have to be open-minded. I can't be closed-minded.
No, but feel free to take a picture.
Yeah, and I like this stuff because I know a little bit about it. I don't know as much as you do, obviously, but it's good to learn more. You know.
Yeah. No, it was a really cool lab to be a part of this summer.
All right. All right. I'll talk to you. I'll see you around. All right. Cool. So we're at the MIT building, and a lot of interesting things out here. See if I can show you guys what we have. We'll take a quick walk around and show you all the stuff that they have. This one's not on again. I think a lot of them are taking a break, so they're going to be shutting these down. But let's go take a quick walk around so you can see.
I mean, if you need any type of if you want.
Very concentrated.
A lot of interesting things out here.
Oh yeah.
Pretty interesting.
So what I've heard is that the first year students are on this side, which means that it's a little less technical because they only have 1 year experience. And then on this side is the second year students, so it's a lot more technical.
Gotcha. I was on the more technical side, so I'm gonna go to the less technical. All right. Thanks. Thanks for the heads up. I'm just gonna show. It's hard to get a good image because there's a lot of people out there. Get the little petri dishes there. Now, what does she have inside there? Do you know?
Well, we're gonna explain it a little bit. We're gonna explain it a little bit.
Okay, go ahead. Yeah.
We're gonna present the sound lab, and my name is Gabby.
My name is Samara. My name is Jocelyn.
So, what is one thing you love to do? I would love to research about like diseases, like how they work in the future, like.
For me, I feel like all the interesting fact that I know of is that engaging in the early learning activities later helps you with stuff such as reading, writing, and so the reason I'm passionate about STEM is because it's something I've always liked ever since elementary school. I always look forward to going to science class and getting to go outdoors and learning about nature. So here's our content overview that we're going to when you guys drop your presentation. Alright, so who is Stan? Not Stan Lee, the Marvel icon. A Stan is a patient who might contain bacteria MRSA on his skin. And we are trying to identify the bacteria present on his skin because he's about to undergo surgery. And we want to treat him for any bacteria he has present on his skin to prevent infection. We are focused on MRSA because MRSA is a methicillin-resistant which is just a type of staph bacteria that is resistant to many antibiotics. And this is a picture of what MRSA, the bacteria, would look like in an MSA bowl. It would turn yellow, and you can see the colonies of bacteria. So this lab was split into 2 days. Day 1, we identified the bacteria present on the skin. And day 2, we identified the antibiotic to use for STAN. So here's our day 1 results. We used a petri dish, which is this dish, and it contains iron, and it was used to treat the bacteria on the MSA plate. This is the plate. This is the MSA plate, and the MSA plate is actually the bacteria that can go many types of bacteria, and the MSA will turn yellow if that's MSA. And here's a reference table. That's the types of bacteria, and it goes on the ending part of the MSA
Okay, so they got silence. So we got the MSA plate and the NA plate. In here, we can see the bacteria that is growing, like it's white. And there's the NA plate here. We can see like 2 colors, like there's yellow color and white color. So you can see a little bit here, like there's yellow color and white. And that means that there's 2 bacteria in this plate growing. Okay, next method, like where we straight out the little bacteria, like choose one dot, and then we like get a new plate, any plate, and then like we like put inside, and then like we wait a little like some wait like 1 day, some hour, like to see if you like the bacteria grow, like visualize put antibiotics inside. If you like, if the bacteria can like be resistant to antibiotics.
So day 2 findings. So we placed the 8 antibiotics onto the petri dish with 1 colony of bacteria, and you guys can see here, these are the 8 different antibiotics. And what we're focused on are the zones of inhibition. Zones of inhibition just means the areas where the bacteria is not growing, meaning that the antibiotic is actually fighting off the bacteria. So we go back to this reference table to compare the diameter of each of these circles to see whether the bacteria is resistant to the antibiotic, intermediate, meaning it's resistant but also susceptible, and susceptible to the antibiotic, meaning that the antibiotic is able to kill off the bacteria. And now we made another table pointing out the different antibiotics, and we focused on the zones of inhibition, like I said earlier, and we wrote down our data. And our data resulted in 4 of the antibiotics were resistant to the bacteria, meaning that the bacteria would still be able to grow even with these antibiotics present. And 3 of the antibiotics are susceptible, meaning the bacteria will be killed off by these 3 antibiotics. And one of the antibiotics was intermediate, so it wouldn't be ideal to give it to Stan because it's resistant but also susceptible.
In conclusion, Stan doesn't have MRSA, but it contains another bacteria that is called Staphylococcus epidermis and Micrococcus luteus.
After testing 8 different antibiotics, we determined that Stan can use 3 different types of antibiotics, which are listed here because the bacteria to be susceptible to these antibiotics.
Okay, so ways we've grown. So an example of something I learned in the lab showing outside of the lab was actually during this lab, Stan lab, where we were learning about antibiotic. My mom got sick and she had to take an antibiotic that we tested here, which was penicillin. And inside the lab, we learned how antibiotics work. So it made me have a deeper understanding of what the antibiotic is actually doing to help my mom.
For me, my favorite day workshop was when we did sleep lab on the sleep lab, which was basically we had to find out whether they were learning or not person. And we used our own DNA, we used a swab and we all around our mouth. And we did that with our PCR and tuberculosis to determine whether we were a learning person, and I was intermediate, so I was cool. And individually, these different places, I had 2 scales. So that's all determined.
It's great something that you tried for the first time at LEAH. Well, my first week I tried to take a test. It's called Markov-Mann, and I wasn't not ready, but I was so I want to do it now. I was so busy, and I learned that you should be patient and try get more experience in the lab to get better.
So some acknowledgements. We would like to thank the wonderful instructors from this program because they really helped us so much throughout the summer. They gave us a deeper understanding of the science we were learning in lab. And we would also like to thank the LEAH staff, Bertha and Wesley, because they just made this whole program fun and interesting. And these are our sponsors. Thank you so much. And thank you for listening. Yeah, thank you for listening.
All right.
Do you guys have any questions?
So which is the best antibiotic in your opinion?
I would say ciprofloxacin.
Ciprofloxacin?
Because you see how the zone of inhibition is big.
Uh-huh.
So yeah, it's able to fight off the bacteria.
And that fights off most bacteria.
Yeah.
Okay. Cool.
Thank you.
All right.
About the W kind of bacteria, we would love to.
Also, you see those germs like bacteria, and some bacteria is actually essential to life. Bacteria decomposes in the ecosystem, so any dead organisms you see around it, they will break it down and create nutrients for the soil.
Okay, so once again, the purpose of this is to provide that
We chose to do 9F and 1492R because it gave us a huge amplicon, so that created a good base and it made sure that our success level will be a little bit more higher because our first primer and our under primer gave us a huge amplicon to amplify our DNA and amplify our gene. I hope that answered your question.
That did. Thank you.
Yes. Yes.
Like it's not just I'm so
Yeah. We know too well.
Yeah, you learned the hard way.
Exactly. We also have some growth curves if you care to.
Yeah.
Okay, so we're so interested in our bacteria, and it has like a cool red pigment. So I was like, let's do more experiments with it. So we conducted some growth curves, and we recorded the optical density of our bacteria over time at different temperatures, and we found a pattern. The pattern we found is that at 30 degrees Celsius, our bacteria's double time was 40 minutes. So every 40 minutes it doubled, and there's at 37 degrees Celsius, which is our body temperature, we found that the doubling time decreased, so it doubled every 35 minutes, it doubled. So that's something interesting we found, and yeah.
That's very cool. Thank you. I'm impressed. I'm excited. You're going to come back next year for the second part.
Hopefully at MIT and everything.
All right. Good job.
Any questions?
Excuse me. Sorry about that.
We also identified our bacteria to be gram-negative, which means it's not naturally competent. So as I was speaking before, our bacteria is not naturally competent. So that means that if we did try to take in a foreign DNA, it wouldn't really be welcoming to the foreign DNA. So we figured this out that it was gram-negative or gram-positive because usually when it's gram-positive, this will be a lot thicker. As you can see here, it's a lot thicker lines. Well, if it's gram-negative, as you can see here, it's a lot thinner. So that's how we figured out if it was gram-negative or gram-positive. And I just want to acknowledge our instructors, Madana and Vanessa. They helped us a lot through this time. Madana, she helped us out a lot more with just science. She got us in shape, and Vanessa was really just there helping. I want to give a great thank you to Enoch as well.
Where's she at over there?
He's my grade inspector, or he was my TA, and he helped us out a lot, and he was pretty inspiring. I know he helped us out a lot, and then thank you to the LEAH staff for our counseling outside of just science, our snacks, and helping us to work to the best of our ability. I would also like to thank our supporters and funders. Without this, this wouldn't necessarily be possible, and we wouldn't really be getting paid, which is practically the best part. And yeah, thank you for listening. Is there any questions?
I have a question.
Yes, sir.
So what do you think went wrong, and why did your ligation not work, and what could you have done differently?
So I feel like where we went wrong, we didn't necessarily know where we went wrong, so we redid our ligation since we needed our ligation to actually perform bacterial transformation, we try to go back and redo our ligation. We try to go back and do our ligation again, and it still didn't work. So from that point on, we just determined that it might have been a process even before that went wrong. But we didn't necessarily have the time to go back and do that much our process again. So we just decided to use our peers and take the help from our peers.
She's smarter. She's smarter or you smarter? She's smarter?
By a mile.
By a mile? Okay.
How many feet are in a mile? Yeah, you gotta 5,280.
Yeah.
Glitching.
She's smarter.
My thing keeps on glitching.
All right. You're smart too. Don't take that away from me. We're all smart in our own way, you know.
Yeah, I'm gonna get smarter.
You will. Well, keep trying. You always get smarter.
That's a fact.
There you go. Never give up. There's a lot of interesting things out here.
We'd like to say thank you to our funders who have supported the share without them. And we wanted to say thank you for listening.
All right.
Any questions?
That's all out here. All right.
So our presentation is on utilizing LEAH and bacterial transformation. My name is Eden. I'm a rising senior at the Heritage of Latin School.
I'm Naveen, and I'm a rising junior, also at LEAH. So oftentimes in biology, you might have some DNA and you want to make copies of it, you want to replicate it. So for us, and for our project, we use bacterial transformation to make copies of DNA. And we did this by adding DNA to bacteria and then growing. And as that bacteria divides, you get 2 copies, divides again, 4, whatnot. And that looks like this, a plate of bacteria. So each of those dots are millions of bacteria all clumped together. So how do you tell, if your transformation is successful? How do you know that your bacteria is actually being cloned? One way you could do this is by sequencing. So you could try, picking every single colony on the plate and testing whether or not it has it. That's really slow. It can take up to weeks to get results. And it's very expensive too. The better option is lactic acid, which is very, very fast. It's effective, it's cheap, and it's very easy to interpret the results. And it's all built upon LEAH gene.
So the LEAH gene is found in the LEAH operon, which is exists in many bacteria. And this operon is basically a collection of genes that work to regulate the metabolism of lactose. And we're more specifically concerned with the LEAH gene. And this operon functions to encode for the enzyme beta-galactosidase or B-gal, and B-gal will break down lactose into two simple sugars, the lactose and glucose, for the bacteria to use as energy. So that's the normal function of the LEAH gene. But during our experiments in the lab, we use it for a different purpose. So we use the B-gal enzyme that the LEAH gene produces to cut a colorless chemical called X-galactosidase or X-gal. And when X-gal is cut, it will form a byproduct of the lactose sugar as well as a chemical that forms the blue pigment. And we'll talk more about the purpose of the blue pigment later.
So how are we going to get this laxing gene working? Well, to be able to add it to our bacteria, we're going to first use something called a plasmid. So a plasmid is just a circular piece of DNA, and this one is called a PGMTP. So on this DNA we have our laxing gene, as discussed earlier, and we also have an ampicillin-resistant gene. So this ampicillin is an antibiotic, so this gene will make bacteria resistant to antibiotics. And because we want to replicate our DNA, so we're going to attach our target DNA this direction. We'll go right in between our lactase. So far, if this was successful, now we're going to add our DNA to the coli cells. This is transformation, so we can start growing them and start that process of purification. And there are a possible couple outcomes from this transformation. So the outcome we want is we have our normal cell, and it has our target DNA sitting right where it's supposed to. There's also another bad outcome where our target DNA is not added. So you can see there's no green, it's just blue here. That's not something we want. We want our target DNA there. it's also possible that the bacteria doesn't connect to any DNA at all, no plasmids, no
So our two blue screening scenarios is our successful scenario where our target DNA gets ligated within the LAC-C gene and disrupts the function of that gene, so it can no longer produce the enzyme BGAL and produce that blue pigment. But our other unsuccessful scenario is that our target DNA doesn't ligate onto the plasmid at all. So the LAC-C gene actually ligates to itself and forms a complete gene, and then is induced and turned on by a chemical called IPPG, and continues to do its functioning of producing that enzyme BGAL that will cut XGAL and produce the blue pigment. So to set up our LAC-C assay, we got an LB agar plate and we put two chemicals, XGAL and IPTG on it, and then also the antibiotic ampicillin. And then we put our E. coli transformation from our transformation tube onto the plate and incubate it in a 37 degrees Celsius warm room to grow overnight. And specifically 37 degrees Celsius because that's what E. coli grows in best. And our results showed a mix of blue and white colonies on our plate.
This is a picture of our actual results here. You can see all of our blue and white colonies. So these blue colonies, now what we want, they don't have the target DNA, just the gene intact, so they produce the blue. And then all those white ones are the ones we do want. Now you might be wondering, okay, well how do we tell our normal E. coli cells from these? Well, because we're adding this ampicillin, this antibiotic to the plates, these normal cells are just going to die. They're going to be killed by the antibiotic. And the reason why these don't survive is because this plasmid contains an antibiotic resistant gene. So only these are going to be able to grow on the plate.
And so our transformation results show that we had a 63.31% transformation efficiency, which is basically the number of successfully transformed white colonies we had out of our total colonies. And this is a pretty good transformation percent, so that indicates that we had a pretty good amount of successfully transformed cells. And to conclude and kind of tie up what we explained to the real world, transformations are a technique that are often used in biotech, medicine, and other fields. For example, the hepatitis B vaccine was created with a yeast cell being transformed with the hepatitis B viral DNA. So it's crucial to have an understanding of LAC-C blue-white screening for someone that's doing a transformation to help determine their transformation's efficiency and success. We want to thank our supporters and funders, as well as our lab TAs, Paloma, Laura, and Enoch. And
our professors, Dr. Chung and Dr. Sassi.
All right.
We have 7 months. And because it was so successful, we didn't need to do it again.
What was the most challenging part?
I'd say, in my opinion, the most challenging part is definitely transformation, because cells don't want to take in foreign DNA, right? So there's a lot of measures against that. So you really have to be like really gentle, and it fails a lot, very often.
All right. Good.
Michelle, we'd also like to thank you. Our flies have been here to use whenever the
Stand in front of your poster 2 seconds and act like you are. I wish you had taken a real picture of your plate because you had nice bacteria. A little closer. No, a little closer. You can grab that part. Alright, excellent.
It's characterized by differences in social communication, cognitive and behavioral flexibility, and conceptual decision. Compared to typically developed individuals, individuals with ASD struggle to with decision making in unstable environments where rules can change unpredictably. And basically, people with ASD tend to move into more repetitive behaviors, hypertrophic behaviors, which is just
Alright guys, I figure I'll show you a little activity going on. A lot of interesting things out here.
So, why are we generating the expression vector, you might ask? To understand that, you will need to understand what our entire process is. I'm currently generating an expression code. However, simultaneously, my PhD student that I'm working with, and also an undergrad student that I'm working with, they're generating a host cell line. That's UQOS, Q93, and NUG-C3. UQOS, it has a high proliferation rate, and is also commonly used in DNA damage repair. Whereas Q93 has a high transfection rate, which is why we're using that for the flip-in system, which uses your FRT sites. You can see your FRT right here. And also an FRT site on your POG44. And using that, you will create your expression host cell line, which is expressed with RFW3 gene in the host cell line, as UQS, NUG-C3, or Q93. And also, we're going to mention NUG-C3 is just control here because it has a mutation in your RFW3 gene. So, if you make changes to your Q93 or UQOS, you expect a different result than NUG-C3. So, now that you have your expression host cell line, you will take that and you will do bio-ID, which is basically similar to ubiquitination, as I mentioned before, will attach a lysine molecule onto different proteins that come within proximity of the RFW3 gene. So, because you have the Bure star, the Bure star is a mutation of the gene Bure, and it will add a lysine group to every single protein that comes within proximity. And using that, you can filter out different proteins that have your lysine group attached onto it. So with your lysine group, you've got your suspect list now. Now it's up to you. This is why here I have bio-ID and more, because there's different things to do after bio-ID. So with bio-ID, you have your suspect list, and now it's up to you to determine how do these proteins interact with RFW3, and how does RFW3 recruit them for DNA damage repair, whatever process they are involved in. That concludes our presentation. Thank you for listening.
If you don't enjoy the training, then it's a loss. But that's why you gotta, there's a little bit of geek in all of us, right? I like
designing an experiment.
Alright guys, thanks for watching. Hope you got a little education off of what these people were explaining. Catch you guys in the next one.